Hi everyone, welcome to this week's lecture for MU418, focusing on viruses and cancer. So for this lecture, I'll be talking about mainly cancer-causing viruses, but also will describe oncolytic viruses and the use of viruses in therapy. So the objectives for this lecture are to differentiate between these viruses that are carcinogenic or cancer-causing and oncolytic, and to describe how oncolytic viruses are used in therapy and assess their efficacy. So the resources relevant to this class, there's a chapter in Cancer Immunology textbook, as well as a review article about oncolytic virus therapy, about the current state of oncolytic virus therapy and upcoming and experimental treatments. And I've also posted a link to a video from iBiology, which is presented by Dr. John Schiller of NCI. And he explains HPV, the infection, and development of the vaccine. So if you're interested, there's those resources. But to begin with, there are many viruses that have established links to cancer. And of these, I'm only going to focus on HPV. So human papillomavirus, there are many strains of this virus, but there are two that are prominent in HPV-related cancers. It's HPV-16 and HPV-18. And the early, I guess the early designs of the vaccine were mainly focused around these two strains, but the latest vaccine product, Gardasil 9, protects against 9 of these HPV types, including low-risk types that cause genital warts, as well as 7 high-risk types. So HPV is the cause of about 5% of all cancers worldwide. And while, you know, most of us are probably familiar with the cervical cancer link to HPV, it's since been found that other cancers of different types are also caused by HPV. And you know, due to the nature of this being a sexually transmitted virus, yeah, you can get lots of different tissues infected. So I want to start with some kind of basic histology to get a sense of how HPV infects and the HPV life cycle within the tissue. So in a normal squamous epithelium, so not sure who's familiar with histology images, but I get my laser pointer. Yeah, we'll demonstrate. Here we go. So this is a biopsy or a section of a biopsy from normal squamous epithelium. Squamous just describes the cell organization. But in this image, you can see it's like a pink and purple stained tissue. This is through the use of a very commonly used, widely used dye or pair of dyes, hematoxylin and eosin staining. So hematoxylin is kind of a blue dye, and it binds to and stains the DNA or nucleic acids. So you can see the nuclei of individual cells. And then the eosin is a counter stain, which stains, I mean, bright pink on its own, but the combination gives this sort of purple color. But eosin stains basically cytoplasm and other cellular structures, so you get highlighting of the nuclei. So in this section, we have the stroma, layers of just stroma underneath the epithelium. And this densely packed region of cells is the basal layer. You can see many nuclei and lots of proliferation. But as you go towards the top of the layers of the epithelium start to lose their nuclei and desquamate and lose their shape essentially, and they will die. So there's continuous replacement of the epithelial layers from the basal layer. They proliferate, differentiate, and eventually die to be replaced. So that's in the normal tissue. So in the case of HPV infection, you get a progression from normal squamous epithelium towards these cervical intraepithelial neoplasms, so CIN. And the range is CIN 1 to 3, but yeah, I've only included 1 and 3 here to see how different this is. So in the early stage, the CIN 1, you can already see, although I know this magnification is different, but you can see that the basal layer is here. So you still have a basal layer intact, but you see much greater numbers of cells and this koilocytosis. So you're getting nucleated cells and aberrant nuclei progressing up through the layers and even up the uppermost layers. So they're not differentiating or de-differentiating, I should say. And so you get greater numbers of cells and disruption of these layers. And then as CIN3 happens, you can see much greater loss of the layers. The basement layer is probably down here, but basically it's all proliferating nucleated cells all the way through. So this is late stage intraepithelial neoplasia. So if this is left untreated, not excised, so on, this will progress to cancer. So to show you again a comparison of the normal squamous epithelium, the nice layers, and again this is a different magnification. This is the basal layer, that's the top, and in this image there's some adjacent normal tissue, so that's the basement layer, and that's the very top. And you can see this carcinoma has formed and it's destroyed the basement layer, basement membrane, and has invaded into the stroma. So you get dysmorphic cell growth, structures, disorganization, and likely invasion into lymph nodes and through blood. So a patient diagnosed with squamous epithelial carcinoma or squamous cell carcinoma, SCC, you might see that abbreviation. They will go on to have surgery to remove the main lesion and possibly involved lymph nodes as well, but they'll also receive a combination of radiotherapy and chemotherapy. So now that you have a overview of the structure of what it looks like in real life, now we can talk about how and where HPV infection occurs. So it's a kind of a highly ordered process, and it goes along with the epithelial differentiation, but it's a very acute process. Within 12 weeks, the virus has infected, replicated, and can go on to further infection. So I'll go through this step by step. So first, the HPV infects at that basal layer. So it doesn't infect in these keratinocytes that are, without a nucleus and desquamating a diet. It really, the exposed basement membrane is where the HPV infects. And then at the next stage, once it's established an infection in that basal layer, it will not, it'll replicate only the necessary genes and start transcription of early genes. So this is just to maintain copies of the HPV DNA and support transcription and replication. And as the infected cell is moving through the epithelial layers up towards the surface, this is when those host cells will stop dividing and start differentiating. And this is when the HPV DNA is able to really replicate its numbers and prepare for further infection. And then finally, right at the uppermost layers, this is when the final gene set are expressed. So the late genes, that these genes expressed, these genes are expressing proteins that form the viral caspid proteins. So those are the proteins that envelope the virus and allow these virons particles to be assembled and complete and ready to be released. And they're released as these cells are replaced. So HPV isn't a lingering infection. It's not, doesn't really lay dormant forever like some viruses do. HPV can be cleared by the immune system and it involves a couple of processes, but the first and most important step is by antigen presentation. So within the epithelial layers of basically every tissue, but most relevant in the cervix are the Langerhans cells. So these are a type of macrophage. They're a highly specific macrophage that are resident within the skin and mucosa, and they're immune sentinels. So they're continuously sampling their microenvironment to look for antigens, and if they are activated by antigen, if they detect antigen, then they leave their tissue to present antigen in the lymph nodes to activate T cells. There are also dendritic cells, but these are present in that stromal layer, the outer layer, not within these layers of the epithelium, and they can basically sound the alarm. So they can secrete these pro-inflammatory cytokines to, alert other immune cells to migrate towards this site. They can also phagocytose pathogens and present antigen to T cells in the lymph nodes. So once an infection is detected or antigen is detected by these immune sentinels, these antigen presenting cells, APCs, present their antigen to T cells within the lymph node that are labeled naive T cells. They're just inactive T cells. This diagram's a little messy, but I'm going to highlight each part as best I can. So here is a dendritic cell in this case presenting antigen. Antigen's the little red peptide being presented to this naive T cell in the lymph node. So upon antigen recognition, the T cells will proliferate and become activated or mature into both CD4, so these are the T helper cells, and CD8, these are the effector T cells, the cytotoxic killer T cells. So the CD8 are the ones that directly kill the infected cells, and CD4 will help by secreting additional cytokines to recruit more immune cells and help with antibody production to neutralize the virus. Yep. And finally, once these T cells are educated, proliferating, ready to go, then they infiltrate to the infected tissue site and cause specific killing of the infected cells. So if this is the case, why is HPV infection causing cancers? Why is it present if we have these expert cells that can detect it? It's because HPV has evolved to be really efficient at evading recognition by the immune cells. So this is by a couple of reasons. One of the major reasons is that it does not cause cytolysis. So some viruses, when they infect a host cell, they proliferate and generate new copies of virus to further infection. They'll cause the cell to die or lyse, and that releases the virus particles. But because HPV, it doesn't need to do that, it gets a free ride, it's released at the uppermost layers of the epithelium where the keratinocytes would be dying and desquamating anyway, so they get released there. Another reason is because HPV also it hides within the host cell. It doesn't cause cytokines to be released. It doesn't alert the sentinels that there are, that there's anything wrong with this infected cell. So it evades that recognition by the antigen presenting cells. So it's kind of like silences the alarm in that case. And another reason is because, again, by being released at the surface of the epithelium rather than the basal layer, within the stroma, indicated that there are, that's where blood supply and lymph drainage is. At the cell surface, this is there's very little blood vessel that reaches all the way to the top, so you don't get detection in the bloodstream, so there's no viremia, so that's like virus within the blood. So it's really very sneaky and has a lot of ways that it can evade recognition by the immune cells. So know about how HPV infects and persists in its life cycle, but how does it actually, cause cancer? It's because of the HPV viral oncoproteins and oncogenome. So HPV integrates its own DNA into the host genome. So cervical epithelial cells or whatever epithelial cells are infected, HPV inserts its genome. And so those, the inserted genome is transcribed, making messenger RNA that translates into these oncoproteins. So that means like cancer-causing proteins called E6 and E7. And these oncoproteins, they're not just for promoting virus survival and so on, they actually interact with the host tumor suppressor proteins particularly P53 and retinoblastoma, RB. These are two major tumor suppressor cell cycle regulating proteins. And so they become inactivated through ubiquitination and other methods. P53 gets degraded. E2F is a different pathway, but retinoblastoma is inhibited. So this E2F transcription can be allowed to go uninhibited. And so these combination results in uncontrolled proliferation and unrestricted entry into S phase, so lots of replication without checkpoints within the cell cycle that would be activated if there's an issue with gene transcription. This would be stopped, and P53 regulates the cell cycle also, so you get uncontrolled proliferation, resistance to cell death, lots of replication. So the E6 and E7 oncoproteins really have a huge influence over the host epithelial cells. This is just the entire picture. It was too large to put in one, I thought. So for interest's sake, I put this slide in because it's interesting to me, at least. When I got here, I got told that, Dr. Doug Lowy and Dr. John Schiller, they're they invented the cervical cancer vaccine. But where I'm from and where I studied, University of Queensland, I was told that Professor Ian Fraser and Dr. Zhan Xiao, they were the inventors. So there's actually 4 recognized claimants to the technology for creating the vaccine. So at NCI, they're well known for their role. This is Dr. Ian Fraser and Dr. Zhan Xiao, since passed. There's also a group at Georgetown and a group at Rochester that were also recognized as creators of the vaccine. So there's an article I also included on Canvas. It's a short article just kind of describing the timeline of how the vaccine was created and so on. And I guess it's just something I like to highlight, to be aware that it takes many, many labs to make an important breakthrough such as this. So I took a screen grab of Happy John Schiller here, describing the HPV vaccine from that video that I also posted. But the vaccine is interesting because it is made of proteins. So it doesn't contain a live virus. It's basically immunization of virus-like particles, so empty virus, essentially. There's no viral DNA and they're not infectious. It's really just protein that is the antigen. So the L1 capsid protein is the antigen. So in this little diagram, there's these proteins that kind of assemble into these pentamers that basically coat the outside of the virus to create an envelope inside of which the HPV genome sits. So by making this the antigen, neutralizing IgG antibodies against that protein are produced. So this means that vaccination protects against infection, not clearance, and not once the virus is established. It prevents virus entry. So for the Gardasil 9 and multivalent vaccines for HPV, they contain different virus-like particles, VLPs, for each HPV type. And so that any differences between HPV 16 and HPV 7 will be covered by the vaccine because they contain the different L1 pentamer proteins. So finally, I'm going to talk about oncolytic viruses a little bit. I know Dave will also be covering this in his final lecture. But oncolytic viruses, they're really interesting. There is only one currently approved oncolytic virus approved for use in cancer therapy by the FDA, although this is an interesting and growing area of research to try and identify new viruses or other ways that we can use these viruses in cancer therapy. So Temilogene leherparavovic, or TVEC, I'm going to call it. Imilgic and Oncovax are brand names. So TVEC is is a genetically modified herpes virus, and it's only been approved for treating melanoma so far. But basically, it's the herpes virus. It's very good at infecting cells, but it has some genes removed that are able to inhibit the immune response. And also, they've inserted a gene that enhances anti-tumor immunity by recruiting the immune cells to that site. So because the herpes virus, unlike HPV, it does cause cell lysis, that means that once the virus infects the tumor cell and lyses, it causes this GM-CSF to be released to call anti-tumor immune cells to the site, but it also releases antigen from the tumor so that those immune cells that are recruited, they can pick up the antigen and present it to T cells. So I wanted to present, excuse me, I wanted to present this clinical trial data from the analysis, the use of this TVEC in unresectable, very aggressive late stage melanoma. So in a clinical trial, they want to demonstrate that, so especially in these patients, this is a cohort of basically limited to no treatment options at this late stage. They want to demonstrate efficacy of the virus, and, of the therapy, and safety, and that it has to be better than the current standard of therapy. So in this stage 3B to stage 4.1 metastatic disease, they demonstrated that treatment with TVEC compared to treatment with the inserted gene product, so GM-CSF on its own, without being released by the virus, they got an ORR, overall response rate, of 31% in these very late stage patients, compared to 6.4% with just the GM-CSF. And duration of response, or durable response rate, it depends on the definition that they use, but basically 20-fold when you have TVEC containing GM-CSF compared to just GM-CSF alone. So this is pretty incredible for late stage disease. And so if you're not familiar with this kind of graph or plot, this is Kaplan-Meier survival analysis. So you plot the outcome of every patient on the trial over usually over months of follow-up, and you plot the percentage of patients surviving over each month. And so in the red line, this is the TVEC patients, TVEC-treated patients, and the blue is the GM-CSF patients. And so the standard is basically 50% survival. So 50% of all patients treated with GM-CSF will survive approximately 20 months, whereas those treated with TVEC will be over 25. And actually, I should just look at this, the median overall survival of all patients treated with TVEC is 23 months. The range, 19 to 29 months, so that's a huge response. Compared to just GM-CSF, the median of all patients was 18.9, which is less than the lowest or shortest response TVEC patient. And the range is 16 to 23. So you get a really durable, strong, statistically significant response in these patients. So that was in all patients. When they looked at just the stage 3B to stage 4A, essentially. So this is stage 4C. So that's, that's, I guess, the worst. 4A is slightly better. When you separate out those few patients that were 4B and 4C, you can see a much more significant difference in the response of survival, getting up to 46 months median survival in the TVEC patients, and the upper limit, it means like not reached. So they're still tracking some patients' survival with TVEC treatment. I also want to show this waterfall plot. So another important way to determine clinical response is not just looking at survival, but looking at duration of response. So this is a little hard to read, but on the left of this, so 0 to 21 on the left, going this way, they've plotted the patients treated with GM-CSF, and I can't recall, but in this, I think they only plotted the patients that had a response, and they classify the response as either a partial response in red, so they might have responded for a certain amount of time, maybe 3 months or 18 months, and partial response generally means either no change in the tumor lesion, so it can be stable disease, or a small reduction in the size of the tumor. Whereas a complete response would be a defined standard of tumor shrinkage. And for the TVEC treated patients, on the right side of this waterfall plot, they have a duration of response that is longer in some of these patients, 0 up to 27 compared to 21. And you can see a lot more patients had a response and a lot more patients had a complete response. So this might be defined as significant tumor shrinkage, often like a level or a percentage of tumor shrinkage is the criteria. So something like 30%. They see greater than 30%, for instance, shrinkage of tumor, then that's a complete response. Whereas a partial response will be less than 30% or stable disease, but they haven't progressed, which is the main thing. But this is a really very impressive response, very impressive study. So I think that's a very positive and exciting area of research that is ongoing. So how TVEC works, and I'm only going to go over this briefly because I know Dave covers it, but basically the lytic function of HSV, as I said, it to release the virus particles, HSV causes cell lysis, so the tumor cells that are infected with the virus get destroyed. But most importantly, it is when the lysis occurs, the GM-CSF is released from these dying cells to recruit mainly dendritic cells and other antigen-presenting cells. And so they come to the site of the GM-CSF and the dead cells, and they pick up tumor antigens. So this creates what is defined as like an in situ vaccine. So the dendritic cells then, full of antigen, go to present them in the lymph node to the T cells to cause a response. The only limitation so far with this kind of therapy is the limitation of access. This can't be given systemically for obvious reasons. It's not targeted, and HSV is quite infectious. So if you gave a systemic dose of this oncolytic virus, it would, lyse any cells. So far you have to inject IT intratumorally or subcutaneously. That's why melanoma was a good option, or this therapy is a good option for melanoma. But hopefully newer therapies can be either targeted or develop ways to deliver this drug or therapy. So in summary, I've shown that there are several viruses with known links to cancer, but I really only focused on HPV, and that the high-risk HPV strains are able to infect different cell types, different tissues, and cause different epithelial cancers. HPV evades the immune system, so it's very efficient at hiding, and also produces oncoproteins to disrupt the cell cycle and enhance cell proliferation. And the HPV vaccine, so it was made to prevent infection, so the virus is not able to enter the cell. Oncolytic viruses, they are, as I showed, an effective therapy, but they're limited so far only by access, and they're an emerging field of therapeutics. TVEC being the only currently approved FDA-approved oncolytic virus for use in melanoma. So to address this week's assessment, I only focused on HPV because I want all of you to focus on a different virus, one of the following viruses listed here, and either describe whether it is an oncogenic virus or carcinogenic virus, what cell type it infects, and how it promotes cancer growth, or if it is an oncolytic virus, describe how it is used in therapy. And it can be, it doesn't have to be approved like TVEC. It could be an experimental preclinical therapy, but please provide references for your, should be about a page and will be submitted online. If you want to talk about a virus that's not here, just send me an email, but any questions, and I'll be happy to answer those. So I'll see you